Hi everyone, welcome to another session of our learning. Today we want to learn about cell cycle, and before we begin we have to understand our objectives of the lesson. So by the end of this lesson the learner should be able to 1. Define cell cycle, describe mitosis and meiosis processes of cell division, and then 3. You discuss the process involved in mitosis and meiosis processes. To begin with, from our introduction, we start with definition of some words. The cell cycle is the entire series of stages that a cell goes through to grow, replicate its DNA, and divide. It consists of several key stages. That is, the interphase stage, where on interphase stage it is the longest phase. That is divided into 3 parts. That is the G1 phase, the S phase, and the G2 phase. We also have the M phase and cytokinesis phase. So in this diagram we have a representation of the 3 phases or the 3 stages of mitosis. The G1 phase, the G2 phase, the synthesis stage, and to crown it all we have the mitosis and the interphase stage. So what is cell division? Cell division specifically refers to the process by which a cell divides into 2 daughter cells. For example, we had one single cell. If we divide this one cell into 2, it becomes 2 daughter cells. Chromosomes are long thread-like structures within the nucleus. Each chromosome is made up of 2 parallel strands called chromatids and connected at one point called the centromere. So we have chromosomes. We have different lengths of chromosome, and at different points we can find the centromeres. That means you can have 3 or 4 types of chromosomes, but depending on where the centromere is placed, we will have a different name for that chromosome. So chromosome occurs in pairs and are known as homologous chromosome. Along the strands of the chromosome, there are genes which are made up of chemical protein substance called DNA. So in that chromosome, along its strand, we have some chemicals which are protein in nature. These chemicals are called genes. And whenever we talk about genes or the genetic materials, we mostly refer to the DNA. So the DNAs are along the strands. So what is mitosis? Our interest today is on mitosis and meiosis. So mitosis, this is a type of cell division which a cell divides into 2 daughter cells having the same number of chromosomes as the parent cell. The behavior of chromosome during mitosis is usually described as a series of stages that include interphase, prophase, metaphase, anaphase, and telophase. So in interphase, these are some of the series of activities that can happen. There is duplication of genetic material, and I've already said whenever we are mentioning genetic materials, we have to understand that genetic materials being referred to are the DNA. And then there's also synthesis of new cell organelles. For example, the formation of the centrioles. Synthesis is just formation. So there will be formation of the centrioles. There will be buildup of enough energy to drive the cell through the whole process. Remember, the process is just beginning. So at interphase, that is the first stage, there must be enough energy. And this energy is in form of ATP, adenosine triphosphate energy. Then we go to the second stage, that is the prophase. So in prophase, the duplicated centrals, remember we said there were formation of new cell organelles. For example, now here we are having the centrals being duplicated, formed again. It was one, but now another form of central is also there. So duplicated centrals separate and move to opposite poles of cell as spindle fiber form. There will be also nuclear membrane breakdown while the nucleolus disappears. The chromosome thicken and then shorten. Then we have the metaphase. So nuclear membrane disappear. Spindle fibers lengthen as the chromosome align themselves at the equator. And there's one thing that we'll notice about metaphase across, whether it is in mitosis, meiosis 1 or meiosis 2, you'll see that the chromosome align themselves at the equator, always at the metaphase stage. In anaphase, that is the fourth stage, chromatids separate at the centromere and migrate to the opposite poles. Remember we had chromosomes, that which aligned in the equator. And now in anaphase, this chromosome separate. When it separates, it becomes chromatid, one chromatid on the other side and one chromatid on this side. So on the opposite sides, we'll have one chromatid each. And then the spindle fibers or the spindle apparatus begin to disappear. And then now in the fifth stage, we have the chromatids collect together at the opposite ends of the spindle, and then nuclear membrane forms around each set of chromatid, and then cytoplasm divides into two, leading to the formation of two daughter cells. Remember we defined mitosis as the formation of two daughter cells. So at the end of this whole process, at the end of the telophase process, we are seeing that the chromatids that were separated, each of them, the nuclear membranes start to form around each set, and then also cytoplasm divides into two, leading to the formation of two daughter cells. Then we go straightly to meiosis. So meiosis is a specialized type of cell division that reduces the chromosome number by half, resulting in 4 genetically distinct daughter cells. So it is essential for sexual reproduction and occurs in two main stages, that is meiosis 1 and meiosis 2. So in meiosis 1, we want to understand what's happening there. So